The process of DNA replication can seem kind of confusing at first, but hopefully I'm going to break it down so that you see how each step of the process occurs, why it's important, and then you're able to pull the whole thing together to understand the whole reason behind DNA replication. First, let's review a little bit about DNA. DNA is made of deoxyribonucleic acid. It contains 4 base pairs, A's, T's, C's, and G's, and those are attached to a sugar and a phosphate group. The sugar is called deoxyribose, and the phosphate is going to provide the energy just like the phosphate groups in ATP provide energy. This is what DNA looks like in a simple diagram. Don't forget, the A's bases go with the T's and G's go with C's. The structure of DNA was discovered in 1953 by James Watson and Francis Crick. However, they weren't the only scientists to be working on what DNA looked like. Other people contributed to the discovery. People like Rosalind Franklin. She made a DNA image through X-ray technology, which looked like this. And Erwin Chargaff found that the number of bases were approximately equal to their bonding partner. This actually allowed us, before anybody could see the molecular structure of DNA, this allowed us to understand that since A and T were found in equal numbers, and then C and G were found in equal numbers, that meant that A must bond with T and C must bond with G. It allows us to form that base pair rule. Cell division is the reason that DNA has to replicate. Think of replication as a copy machine. You have to go through a process in order to make a copy of a piece of paper, and your cells have to go through a process to make the copy of DNA before it can divide. We know cells divide for the purpose of growth and reproduction, repair, etc., and to replace old cells. So you see here, you start as one cell, eventually you become multiple cells. But before the cells divide, they have to double their cell structures, their organelles, and their genetic information. This is the DNA. We learned when we talked about mitosis and meiosis that when you start with a parent cell, you have a bunch of DNA in the nucleus of that cell. When that DNA replicates or copies, the cell is able to split the DNA, the copied DNA from the original DNA, and put the same exact set of DNA in each new cell. So, DNA replication. This is the process by which DNA makes a copy of itself. Think of replication as copier. When replication occurs, you start with 2 sides of the ladder of DNA. Remember, normally this ladder is twisted like a Twizzler. But when replication occurs, the ladder untwists, and then it splits down the middle, and then new bases, the A, T, C, and G, are added to each side, and so you end up with half of the new ladder being from the old material, and half of it being from new bases, and then you have another copy of the exact same thing. When you've got half from the old ladder and half becoming the new ladder, it's called semi-conservative, which means half of the old strand is saved in order to be a template for the new bases that attach. And those bases, remember, attach using the base pair rule. A's with T's and C's with G's. DNA replication starts like this. The ladder untwists. You notice that it starts with like this, and then it untwists. This orange Pac-Man represents an enzyme called helicase. Unwind the DNA. Using the helicase enzyme, which is what unwinds part of the DNA, located here and here. You actually have 2 enzymes, and they unwind or unzip the DNA in both directions. And this unwinding has to be stabilized by some single-stranded binding proteins, just some proteins that would have been made by DNA in the beginning, and they're represented by these three greenish blue dots on the side. And the part where the DNA is actually splitting apart is called the replication fork. It's like a fork in the road, left or right. This is the first step of replication. So to see that again, this orange molecule or orange Pac-Man shaped thing represents helicase. Helicase actually goes through and unzips the bonds between all of the bases, so you end up with two halves of the ladder, one on each side. In the second step, this is where you actually build the daughter DNA strand. You have to add new complementary bases to the old half of the ladder, which is over here. This is also done with an enzyme. The enzyme's called DNA polymerase 3. And DNA polymerase 3 will go through and it will add the correct base to each of the other bases, bonding them together. However, we're missing something. Where is the energy coming from for the bonding? The energy of replication usually comes from ATP, which is kind of like a modified nucleotide. We have a base, we have a sugar, and we have the phosphate groups. When you break off the last phosphate, you get ADP, and then that also releases energy. You remember this. Are there other ways to get energy out of ATP? Sure. If you take off two phosphates at the end, you get AMP, or adenosine monophosphate. This structure here looks just like any one of our normal nucleotides that form part of DNA. Adenine, sugar, and phosphate. So the nucleotides come with their own energy, and they leave behind the nucleotide. Other energy nucleotides, of course. Each of the other bases, guanine, right here a G, thymine, and cytosine all provide their own energy to bond themselves to the old strand of DNA. So here's DNA replication in all three parts. First you unwind, and then the helicase enzyme breaks the ladder apart or unzips it. So you have space to add the new nucleotide bases. Then DNA polymerase 3, which is this purple structure, adds the nucleotide bases in both directions. One DNA polymerase 3 will be going this direction, and another one will be going this way. You end up then with two identical DNA molecules that have an original strand of DNA and then a newly assembled DNA strand. This is semi-conservative replication because it keeps half of the original DNA molecule. This is what DNA polymerase actually looks like. It kind of resembles a human right hand with a finger and a thumb. And the template DNA gets threaded through the palm, and the thumb and the fingers wrap around the DNA, and then the other enzymes work to unzip the DNA and then add the new bases. But there are other DNA polymerases. You've got DNA polymerase 3, which we just talked about. This goes extremely quickly. It adds 1,000 bases per second. It's the main DNA builder in our bodies. However, because it's going so quickly, there is a lot of possibility for error. So we have another enzyme called DNA polymerase 1, which only works at about 20 bases per second, and its job is for editing, repairing, and removing things called primers. On an interesting note, DNA polymerase 1, it was named 1 because it was the first one found, was found by Arthur Kornberg in 1959, and DNA polymerase 3 was found by his son Roger Kornberg in 2006. So this editing and proofreading process, 1,000 bases per second, like I said, means a lot of typos, a lot of errors. DNA polymerase 1 is what proofreads and corrects those errors. It repairs bases that get mismatched, like for example if an A were to accidentally get bonded with a G instead of a T. It removes abnormal bases, things that for some reason didn't form correctly. And DNA polymerase 1 repairs damage throughout the life of the cell. It actually reduces the error rate from 1 in 10,000 to 1 in 100 million bases. That's a lot. That's a big reduction in errors. If you can't picture this, 1 in 10,000 is a lot of errors. And remember that any error happening in your DNA can cause genetic disorders, genetic diseases. It can cause your body to not function properly. So it's important that your body has this proofreading process in order to eliminate these multiple possible errors. So to take a look at what DNA replication looks like, this is the replication fork. The direction of replication is happening this way. This is called the leading strand. However, on the other side, replication is also occurring. That's called the lagging strand, because what happens is the enzyme, DNA polymerase 3, can't just go from one base to the next. It actually has to go a little bit and then pause. And then it has to go a little bit and pause again. And then go a little bit further and then pause again. Helicase, again, is what unzips the DNA. Those are your single-stranded binding proteins that stabilize things. Primase, this only happens on this side of the replication fork. And that, again, is what kind of starts that process. This one has to go in chunks. DNA polymerase 3, working on both sides. And because the lagging strand has to work in chunks, go a little bit and then back up, go a little bit and then back up, those are called Okazaki fragments. And the ligase is what continues to kind of bind those chunks together. Polymerase 1 is proofreading the process, and you have the leading strand and the lagging strand. So let's take a look at what this looks like in kind of all together.
To make perfect copies of our DNA, an enzyme first unwinds the two strands of the molecule by separating its base pairs. Another enzyme then attaches free nucleotides to the exposed sites. The existing strand determines the composition of the new strand. According to the base pairing rules, adenine bonds with thymine and guanine with cytosine.
This thing working here, that is the DNA polymerase 3.
In long strands of DNA, replication occurs in many regions simultaneously, forming the sister chromatids required for cell division.
The next part of the process is to use a strand of DNA to make RNA. We'll just briefly go over this part of the process, and then you'll have another assignment where this is broken down further. Remember that RNA is used to make proteins. RNA is the messenger that takes the code out to the ribosomes, and then those ribosomes make the proteins that control everything about the organism. DNA goes to RNA, RNA goes to protein. DNA gets replicated here in the cell's nucleus. The messenger RNA carries the code out to the ribosome. The ribosome is the factory that makes the chain of protein. These processes are called transcription, which is where RNA is made from DNA, and then translation, which is where the protein are made from the RNA. Just a quick view of what that looks like.
The molecule DNA contains the information to make proteins. In the first step of protein synthesis, a length of DNA unzips. Now nucleotides float in and form a strand of messenger RNA. The molecule of mRNA frees itself from the DNA and moves out into the cytoplasm. Here, the code is read by large molecules called ribosomes. Strings of amino acids link up to make complex proteins.
So we'll go over transcription and translation in another assignment. If you have any things that you didn't understand, if there are any questions about your assignment, please don't hesitate to message me.